This video is about transcription. Transcription is the first in a two-step process that we might call gene expression, where a gene becomes a protein. This process starts with DNA, because genes are made out of DNA. And the process we're talking about with transcription is taking that DNA and using it as a template or information to make RNA. The process after that takes that RNA message and makes the protein, which eventually leads to your phenotype. So we're going to look at this process here of transcription. Transcription starts with the DNA inside the nucleus, and we want to keep the DNA safe and sound in the nucleus. It is our instruction manual for the cell, and we want to be able to keep it there safe and sound. We don't want to be taking it out of the nucleus where it might get damaged. And so we're going to transcribe that into an RNA message, which can leave the nucleus and go out into the cell. And it's going to go to one of these little red dots, either free-floating or on the rough ER, which is the ribosome, and that ribosome is where the next step, translation, will happen. So we're just trying to get the DNA made into RNA, which can carry this message to a ribosome to make the protein. So that's its overall purpose. Now, we're not going to take the entire piece of DNA, the entire chromosome or all the DNA. We're just going to take usually one gene at a time. That gene is a segment of the chromosome or the whole DNA, and make a small piece of RNA out of that. So this process works similar to replication in that we're matching up nucleotides with complementary nucleotides. The main rule that's different here is that if this over here is DNA and this is RNA, then instead of T for thymine, we use a U for uracil. So anytime that DNA is an adenine, we're going to match it up with a uracil, the U, instead of a T. Other than that, the complementary bases work exactly the same. Most of those nitrogen bases are exactly the same as their DNA counterparts, just not that one. Alright, so with that rule in mind, let's take our piece of DNA. Now, DNA is double-stranded, RNA is not. RNA is single-stranded, and so we just need to choose one of these two in order to make a piece of RNA. So the enzyme here is RNA polymerase. Looks a whole lot like DNA polymerase, but this one makes RNA. It does the job of splitting up the DNA, or the DNA itself, and in this case, we're just going to use this strand. So using those complementary base pairing rules, we're going to, again, since this enzyme only knows how to work in one specific way, we're going to start at the 3' end and move down toward the 5' end. And in doing that, we're going to make a piece of RNA. So complementary to C is G, and complementary to A is not T this time. Remember, it is U for uracil. The RNA polymerase, make sure that those are covalently bound so that this whole strand sticks together. Complementary to T, we have A, U, G, C, C, G, A, U. So pretty simple process overall, and this would keep going and going probably for a couple hundred nucleotides at least. And now we have our piece of RNA. There's a couple of different RNA types we could make. So this RNA, once we're done, by the way, we can take this away. We can put our DNA back together, which would stay in the nucleus, so it's nice and safe and sound. So let's put it tuck it away, we're going to now ship this out of the nucleus. This RNA can be one of a few varieties. Main ones we'll talk about are mRNA for messenger RNA, or tRNA for transfer RNA, the one that carries amino acids. Both of those types are made through the process of transcription. There are some other things that happen right after transcription that I might mention, some processing that occurs just in eukaryotes, and one of those processes is to cut out bits of the RNA. This strand may be extremely long, and there are bits that are called introns. Sorry, a bit sloppy there. Those introns are segments of RNA that we don't need. So let's say this whole thing is an intron. We're going to clip that out, make sure that the G is bound to this U, skip over all this, and cut it out. So that's one of the bits. The other one is they get a tail on the RNA and a cap. So on each end they get a tail and a cap, and so the cap goes on this 5' end, tail on the 3' end, and that essentially signals the cell that this is a mature piece of messenger RNA all ready to go and function. It's been proofread and checked by the nucleus, and now we're all set. So this process, remember, of transcription happens in the nucleus, because that's where the DNA is. When it's done, that RNA is going to leave the nucleus through one of these pores and come out to either the rough ER or free ribosomes to do the next process, which is translation.